right. Welcome everybody to Paleo Talks. We are now on episode 26 for this series, if I'm counting right, David.
Yep, that's right.
And today our special guest is Dr. Stephen Josinski. Hey, Stephen.
Hi there.
And today we're going to talk about turtles, and we'll get more into that in just a minute, and a little bit of his background and history. Before we do that, we'll jump over to David to go over how the show works.
Yep, same as we have been doing it for a while now. We're going to start with a discussion with the guest, and then Stephen's going to go ahead and give us his presentation all about turtles, and then about halfway through the program, so 30 minutes in or so, we're going to open it up to questions from the audience. Most of our audience is here on Facebook, so if you're here, leave your questions in the comments when the time comes. If you can't leave a question in the comments on Facebook for some reason, you can go over to the Gray Fossil site, Twitter or Instagram. We'll be keeping an eye on that, and we'll see how many people's questions we can get through until we hit about an hour.
All right, thank you, David. We also have Dr. Chris Widga, who's now joining us every week with adding in more flavor to our conversations. Thanks for joining us, Chris.
Flavor, I like that. I'm flavor.
And I'm Blaine Schubert here at the Center of Excellence in Paleontology at East Tennessee State University, where we also represent the Gray Fossil Site. And actually some of the turtles we're going to be talking about today are the Gray Fossil Site, so we will cover some specific aspects of that. David has one of the shells there. And as I mentioned earlier, these two yahoos, as they sometimes are, we're co-students together here at ETSU. And so they go back a long ways as well. So Stephen, let's go ahead and go to your first slide, and then we will talk a little bit more about what your presentation is going to be about and some of your background. So if you could pull that up. So the first thing I have for you, Stephen, is I have an identification for you on my mug that you have to work your way through.
Okay. It's a turtle.
Yeah, that's good enough. It's got flippers.
I'd say it looked kind of sea-esque, but we'll go with that.
Yeah, we'll go with generic sea turtle.
Simple. That's all the farther.
Yeah, so today he's going to be talking about various turtle projects, including part of your master's thesis when you were here. And so if you wouldn't mind just introducing yourself a little bit more, leave it on this slide. How did you become a paleontologist in the first place? And then what led you into working on turtles?
Certainly. So I started out, I really spent a lot of time outdoors, and so I love animals and nature and everything like that. And I was always really pulled into that. And as a young boy, as many young boys seem to be, I was very interested in dinosaurs. That seems to be a common thing for a lot of people. And most people kind of work their way out of it and grow out of it. And I suppose to a point I did as well. Starting out in college, I went through all kinds of various majors, but eventually I worked out that I didn't like any of those things. I made my way back. And so I got my bachelor's from Penn State, where I started working on mostly early dinosaurs from the Triassic, along with a couple of Cenozoic projects and things like that. But in getting my master's, I moved down to East Tennessee State University, and there was no dinosaurs to work on down there. And so I switched to another group of animals, a modern group of animals that I've always really, really loved, but had never really thought about doing research on, which was turtles. And so I moved to working on and looking at the turtles at the Gray Fossil Site, and especially the one that seemed to have the most material, which was the Trachemes, or the slider group. I eventually went on and finished up with my doctorate at the University of Pennsylvania, looking at dinosaurs again with Peter Dodson. But in addition to looking at dinosaurs, part of my dissertation was still looking at more turtles and turtle evolution, especially Cenozoic turtles. So that kind of pushed me more into learning even more about these turtles and this group of turtles especially.
And on your first slide here, it says you're at the State Museum. So that's a curator position that you have there?
Yeah, so I'm the head of the Department of Paleontology and Geology here at the State Museum of Pennsylvania. And most of our fossil record is much, much older, namely, pre-Mesozoic material, especially pre-Permian. But this is also where I grew up, so it's things I'm really more accustomed to and areas I'm really more akin to. So it also allows me to do field work, normally not with these things I'm going to be talking about today, normally much older things.
Great. Thank you. As I looked at this first picture that you're going to show us, actually, when I first saw that image on the lower right, I didn't even recognize that as a turtle. It's a very good drawing, but at first I thought it was a map. So you might want to actually mention what we're looking at there.
Certainly. So that's a reconstruction of the skull, and the gray area are parts that are missing, and the white area are parts that are preserved. But it's one of those things where most people, when you think about a turtle, you think about the shell and you don't think about the other things, especially skulls of turtles are not exactly commonly viewed. And so you're looking at a side view of the skull of this turtle, namely the slider turtle. The circle to the right is the orbit. That's where the eye would be. And the back of the skull is to the left, where you get an elongation of the squamosal. That's the back top part of the skull that kind of gives the turtle its characteristic skull shape.
So turtles are such an unusual group of vertebrates, compared to many of them. And so as you move forward through your slides, if you could talk about some of the unusual features of turtles, because I know you have a few slides here that show different types of turtles.
Certainly, and it will be kind of telling to then show why turtles are so different and yet so similar with the features that kind of make them the way they are. And
I want to remind you too to do speak up a little bit. We're
Certainly.
Still getting you a little bit low, so if you could get closer to your mic, that'd be great.
It's like somewhat closer without too much of my face, right?
Perfect.
We don't want to see that kind of thing. So
okay.
That works.
You're very small on our screen, so.
Okay, that works. Okay, so we're good?
Yep.
Okay. So in looking at turtles, again, the idea is that when you see turtles, you know what a turtle is when you see it, even if they come in various shapes. And so no matter what you're looking at, it's very easy to tell that it is a turtle. And when you're looking at turtles, they come in a lot of different groups. But again, no matter what you're seeing, you're going to know that a turtle is a turtle. And even though you get some inconsistencies and some weird ones, a lot of the features are still going to be similar and the same. And some of those features show you that even with things that you have on them like shells, turtles can still live in a variety of areas. So you have a lot of terrestrial turtles. You have, of course, the fully aquatic marine turtles. You have semi-aquatic turtles that are living in both areas. And they're going to be able to move in areas even with their weird features that make them turtles and allow them to be okay in all of these different scenarios. But what makes a turtle, at least in our mind from what we see, is the shell. And so the shell is very characteristic. And again, it's one of those things where looking at the shell, it's not something the turtle lives in, it's part of the turtle. So here on the right, you can see that the bones are basically part of the turtle itself. It's not a turtle living within the shell is part of that turtle. And
another thing, David, to point out with that slide, if you go back for a second, that people don't realize, and then it's kind of hard to get your head around it when you first realize it, is that the limbs are on the inside of the ribs.
Yes. And so the outer part of the shell seen here at the top are the ribs itself that have kind of grown and fused together to form this armor shield around the body, but the limbs themselves have now been moved into the shell as well to allow further protection. It hurts mobility in many ways, but it's one of those things that we don't see that in other animals. It's an extreme morphology.
Thank you.
And so when you look at the shell is bone itself, but it's also covered, and it's covered in these keratinized scutes. And they're essentially, they're keratinized or keratin like a lot of other keratinized things, similar to horn sheaths and things like that you see in bovids. But it gives the shell a lot of color. It also helps add in something extra to the shell to give you more morphology when you're looking at these turtles, to be able to identify them more easily, because those scutes are not always going to be the exact same way. The morphology, the shape is going to change between turtles. So when you're looking at the shell itself, the shell is made up of two major parts. You have the carapace on top and the plastron on bottom. And so these are two distinct things, and then they fuse through sutures at the bridge, which is that area in between that kind of protects the sides and holds the shell together. When you look at the carapace, again, on the left, you'll see that it's separated into various bones, and these bones all fuse together to create the shell. On the right, you'll see the scutes, and so the scutes are separated by sutures. So even if they're not there, the sutures, the pieces along the side that show the outer morphology of those and where they lie, are still going to be present. This is present on the carapace, but it's also present on the plastron. So again, the bones and the difference in bones are shown on the left, and then the scutes are shown on the right.
And you should be able to show that with your pointer, David.
Yeah, so
Stephen, see here, I just did it again. Stephen and David. Yeah.
Bones here are on the left, and the scutes are on the right, and the darker lines are separating those. But this gives us a lot of morphology to identify and separate out turtles. So it means that even smaller portions of the shell can be used to give us a lot more information. But in addition to the shell, of course, turtles are not made up of just a shell. There's a lot of other things. And so you can see up here in the upper left, these are individual pieces of a shell. And oftentimes that's what we get. But of course, you also have things like the skull itself or cranium. You have vertebrae, these being cervicals, and you have postcrania, here being humerus and femur, that allow you to get more information from turtles and help identify further from smaller pieces of the remains that you might end up getting. But what we're focusing on here is the Amididae. And so that's the focus right now, and they are commonly called pond or box turtles. They're mainly in the New World, but so they're the most diverse and widespread family of New World turtles. Currently, they include 10 to potentially 12, depending on which study you're looking at, modern genera, and approximately 50 modern species, with one genus with 2 species being known in the Old World, namely Europe, and that being Emmys. So the family itself is separated into 2 subfamilies. Here on the left, you'll see the Amididae, and the most famous one that most people will know is the box turtle down here to the right in this group. But there's various other members with certainly a lot of similarities. On the right,
can you give us some of the common names while we're looking at them here, Steve?
Sure. So you have things like the wood turtle, you have things like the northern Pacific pond turtle, the European pond turtle, the Landings turtle, and the spotted turtle. On the right, you'll have the Diversification, that's the other subfamily. And these members tend to be, for the most part, a bit larger and more semi-aquatic or aquatic, depending on how you look at that. You have things like the chicken turtle, the painted turtle, river cooters or cooter turtles, diamondback terrapins, map turtles, and slider turtles. And so slider turtles, being trachemes, are the main one that we are thinking about currently. Early on, there was not a lot of effort put forward to deciding what the relationships of these groups are, but here is kind of the earliest look phylogenetically at what the relationships may have been from the 50s. And you'll see that the Diversification are over here, and the Amididae are over here. Some weird things that they had set up was that they assumed that they came from a tortoise ancestor, the Testudinidae, with Blanding's turtle being set up in the wrong subfamily, at least from what we know, and a couple members from not even the right family being shown over here in Geomida and Acadia. Now, later on, this is from a couple years back now. This is one of the latest attempts at the genetic relationships of these through a phylogenetic analysis. And we still have the same 2 subfamilies being separated very clearly, with the Amididae being up here and the Diversification being down here. They still separate out well.
So Stephen, how does it compare when you look at sort of the genetic phylogenies, what we're looking at here, compared to the morphological phylogenies? Do they work well together?
So there's, for the most part, they agree for the majority, but there are certain inconsistencies that we end up running into. Certainly things along the lines of where, like say the chicken turtle here, Dirocheles, is considered the basalmost member of the Dirochilines, partially why the subfamily got its name. But morphologically, Chrysomes tends to come out as sister to the other members. So there's a bit of inconsistency there. And the fossils suggest that the Chrysomes is also potentially basalmost within the family. There's a little bit of inconsistency there. There's also inconsistency in where the basalmost member of the imidines comes out. Oftentimes, Clemys is listed as sister to terrapene, but it comes out as far more basal, morphologically speaking, if you look at the entire subfamily. So they're mostly in agreement with a couple of inconsistencies, mostly with who the basalmost member of those subfamilies ends up being morphologically. So if we look at the Immidids fossil record, currently it dates back to around 35 million years ago with Chrysomes potentially not being Chrysomes, but as it was named Chrysomes antiqua from the Middle Shadronian in South Dakota. And the part of the problem is that some members that were considered Immidids early on have been shifted out of the family. So there's been some inconsistency, especially with members like one called Ecmatemes, which is now considered a geoimmidid, the sister subfamily to the Immidids. But most Immitid fossils are still only known from fragments. Even with studies currently putting the divergence date for Immidids in the late Eocene at about 40, which is a little bit older than what we have for fossils, regardless, we don't have good fossil evidence for them going back that far yet. Crown members, or at least members of the modern genera, seem to show up in the Miocene and into the Pliocene, and that's when they really tend to diverge and take off. And we're kind of seeing that a little bit with the fossil record. If we look at fossil Immidids through time, on the bottom you'll see number of taxa named per year, and then you have a kind of count as you're going through time in orange above that. And so you'll notice some areas of peaks. Clearly here, there's a massive peak in when we had a lot of fossil taxa named around the turn of the century. That was mainly because of a couple workers, namely Cope and Hay, who were naming things like crazy at that time and named a whole lot. As we move forward through time, you get a smattering of them through the early 1900s. That's oftentimes where you get a lot of fragmentary things being named from the Midwestern United States and the Southeastern United States. You move into the 60s and 70s, and you get more work being done in Florida and the Southeast. And so we have several Immidid taxa being named at that time. And then we get a couple random taxa being named from various places throughout North America, mainly Midwestern and Eastern North America throughout this time as we move forward. But you'll see mostly there hasn't been, other than a couple peaks early on, it's just been kind of a smattering with a couple of low zones.
And what would you say the proportion is of this being based on shells?
So almost all taxa are based off, almost completely off shells. In fact, I believe only one Immidid is known solely from non-shell material. And that was named in 2012, and it was a fossil species of Graptomes, or the map turtle. A couple species have cranial material, but most of the time it's very, very poor, and I believe it's only two other ones. At least for type specimens that actually have cranial material that go with that specimen itself.
So in many cases, you know, if a skull is found, you wouldn't necessarily know what kind of shell it would have and vice versa.
Correct. That seems to be a problem with turtles in general. When turtles fossilize, what we tend to see is that you only get either cranial material alone or you get shell material, and it seems to be because as a turtle dies and as it decays and goes through it, all the taphonomy that these fossils go through, oftentimes water kind of flows through the shell and removes everything out of the shell. So the skull and the legs and the tail and everything else get pushed away through fluvial and water sources, and so you get the shell remaining, or you'll get a random isolated skull oftentimes, or partial skull, that are found separately. And so knowing how they go together is oftentimes very difficult. Sometimes the only time that you get them together is when the skull happens to be fossilized within the shell and stick in there.
Okay.
So here you'll see a smattering of fossil Immidids, and you get the earliest fossil Immidid currently, Emys euglypha or Trichomes euglypha here, which is just an entoplastron, the front part of the plastron. But oftentimes you get isolated parts of the shell, and sometimes you'll get the shell itself with some of the nicer specimens, but even so, the shell is nicer, but it's very much incomplete still. And so if we focus on the gray fossil site, I won't talk too much about it because many people currently listening or directly part of this are well aware of it. In Eastern Tennessee, you're getting a locality in, again, Eastern Tennessee that's from the earliest Pliocene and probably the earliest Blancan, and you're getting a sinkhole / pond type environment. So it's going to make sense to get a lot of turtle fossils out of an environment like this. The turtles end up being some of the most common fossils that you're collecting, and you get all kinds of turtles in various stages and various levels of preservation as far as completeness is concerned. When you get lots of things like more complete shells or incomplete shells or cranial and skull material. So in focusing on the slider one, that's Trachemys haugrudi, and that was one that I was able to work on for my master's degree while at ETSU, and one that I was able to name after the head preparator, Sean Haugrud, who works there. So this is the holotype shell, and you get a very complete shell, missing a couple minor pieces, but nothing too extreme. And it shows you then, again, you look at the bones here on the left being separated and the scutes here on the right, you get a very complete view of this turtle. Again, with the plastron, you're seeing the same thing, where you're seeing the bones here on the left and the scutes here on the right, a very complete view of what this turtle would have looked like. We're able to use features of the shell, narrow down and show what it was. And that's how we're able to determine partially, again, where this turtle is in the big scheme of things. There's features of the shell that tell you that it's from the Amidids. There's also other features, especially of the plastron, that tell you that's part of the subfamily, the Dirochiliinae. So in combining that, we look at one of the skulls that we have of this, and this over here on the left is the skull. It doesn't look nearly as nice because you get the top part of the skull here and you get various views of the bottom part of the skull down here. And so in looking at that, what we have is when we put it all together, we get a more complete view of what the skull actually was and looked like, and see how it compares with other members of this subfamily, and especially from features of the skull, including the depth of the skull, so the depth being the height up and down, and features of the lower jaw, we're easily able to see that's part of the genus Trachemys, rather than some of the other more similar ones like Pseudomys. So in addition to a really nice skull and the shell itself, we also get lots of postcrania, which is really, really nice compared to what we normally find with turtles, which is very little or isolated. And so you get things like the cervical vertebrae out of the neck, you get things like the dorsal vertebrae from the back, which is within the shell itself, or you get caudal vertebrae from the tail, and these help tell us a lot more about this animal. But in addition to that, we're also getting a lot of postcrania. So what you're seeing are various elements within this that make this potentially one of the best preserved turtles that we know. And certainly when I was able to work on this, I was able to describe all of these elements in great detail, and definitely making it the best described Amidid known, be that morphologically at least, and osteologically, better than any of the modern Amidids that we have today. And so you're seeing various elements from the forelimb up here, and you're seeing things as simple as carpal, so small, tiny bones within the hand itself, or the manus. But you're also seeing various other parts of the manus itself, including claws. You're seeing parts of the hind limb and the pelvis, and you're seeing things like the hind claws. You're seeing things like the astragalus as well. So you're seeing all kinds of parts, and we have the majority of this turtle then that we're able to describe thoroughly and get a good idea. Now, postcranially, there's not a lot of difference in these turtles, and so most of these elements look very, very similar to modern slider turtles or Trachemys. That's not surprising. The postcrania is telling us a lot more about how this animal is living, and this animal seems to be living very similar to modern slider turtles. So in a very semi-aquatic way, it's living a lot in the water, it's coming up on the land, it's basking, it's sunning itself, it's doing hunting and things like that within the water itself. So not surprising, the postcrania is telling us that this lived a lot like the modern Trachemys, and then the shell and skull are helping tell us that it's different than other Trachemys that we know.
And so,
Now,
Stephen, if we could talk just for a minute about difference, and maybe that's what you're going to go into here, but would you say that Trachemys is relatively conservative in terms of its evolution?
So part of what we deal with some turtles, and Trachemys is one of the highest ones in the sense, is that it's conservative, but it's also very plastic in what it's able to do, and it's plastic in its ability to live in many areas. Trachemys, as red-eared sliders especially, are some of the most invasive animals known today because They're pets, they're released, and they're able to live in a lot of different areas where they're released at. So they become invasive all over the world. And not only is that then behavioral, but it's also morphologic. So we're seeing a lot of variation within the modern group, and that variation can make it very difficult to determine what the normal or average of a species is. And so what you consider the normal for that species, rather than what you consider more aberrant or kind of extreme members of a particular group. So in looking at, again, the gray trachemes, what we're looking at is we have a lot of shells, and this is just a smattering of some of the shells certainly that we have. And you have things like the holotype, but you have things like, complete shells as well, and everything in between. And these are just some of the specimens. I believe 2 years ago, I had an MNI of about 3 dozen or so, and I am sure that the MNI, so the minimum number of individuals, is certainly more than that now. And if you look at just total specimens identified as trachemes, not minimum number of individuals, it's far, far higher than that. So we have a lot of material to work with in various sizes, and you get a much better idea of variation within this species, and then I guess variation for the normalcy of what you're seeing with that. So part of the problem in looking at that, though, is determining what the average is. And so not thinking about what the extreme versions are. And you also have to be aware of ontogenic variation, so changes through the animal's lifespan, through the natural history of an animal. And so you get very young individuals. Things like this shell right here is very young. You won't be able to see it, unfortunately, but the costals were not fully sutured to the peripherals here. You can see potentially small little areas where the costals had attached, but not fully sutured to the shell. But the plastron is complete. And so it's kind of filled itself in here. And so it's a young individual, but not incredibly young. But we have plastra, the bottom part of the shell, that still have openings in various spots that show you that the shell was still growing and hadn't fully come together yet. So we have a lot of variation present. So what we need to see is how that kind of works with what we're looking at. And it's important to understand what that variation is. And so you concentrate on the parts of the animal that you know are more important. So here you have things like the shell itself and the skull, and you can compare those and the various parts of those in a more consistent basis. But again, looking at this is just the nuchal, the front part of the shell, the front main bone in the middle of the shell. And it's oftentimes been used in the past to determine differences. And here you might see some differences, but you also see a lot of similarities. These are 4 different fossil species of trachemes. And here is Haugrud. So here's the gray fossil site trachemes, and you'll see that there's a lot of similarity. So there's a lot of similarity between what we're seeing here with only minor differences. And so you have to get an idea then of what the variation is within the modern red-eared slider. And in order to do that, what I ended up doing was doing more of a 90-10 scenario when I looked at the fossils. With sample sizes as large as I had, the idea was to make sure that single weird individuals didn't push the data set in one way or another when I'm looking at normal features for these animals. And so what I ended up doing was making sure that at least 90% of a set, a taxon, a species, had a particular feature. And so if 10% had a abnormality within that, I determined that was not normal for the species itself. Now, this is one way of doing it. It's not the only way, and some people would not agree with that. Some people would determine that if any individual of a species is different, then you should look at it differently and score or determine that feature is different or more widespread within the species. But this was the best way I could think of to deal with the amount of variation you're oftentimes seeing in slider turtles and trachemes because of the amount of variation you're getting. And even with a sample size then, at least in the several dozen for Haugrud, I was able to still determine with some degree of variation where the normal position of that species is going to be, morphologically speaking, within the features that you're seeing. It was able to allow me to place this turtle in a, I believe, a more accurate position phylogenetically within the evolutionary relationships of that turtle. And so here you'll see in red the species Trachemes haugrudi, and you'll see where it landed phylogenetically within this analysis. And as a note Of course, these mean that it's a fossil species. So you'll see a lot of fossil species up here, basal. This is mainly the Dirochiliinae as a subfamily. And you'll see slider turtles here. You'll see map turtles here, sister to the diamondback terrapin. You'll see cooter turtles here. You have the chicken turtle here, and you have the painted turtle up here. And the gray fossil site Trachomys comes out as sister to two Florida species. And these two species up here are from the Midwest, Hilii and Idahoensis. So what you end up seeing, at least currently, is that these fossil species are not grouping with any of the modern species of Trachomys, even though they do come out within the genus. But you get a biogeographic grouping of these animals where you get a southeastern set coming out here together, and you get a midwestern set coming out here together. And this is suggesting that these two lineages were separated out and they were able to evolve independently in two different areas before this time, presumably sometime during the Hemphillian, because you're getting late Hemphillian members of both groups. And so, again, you're getting the modern genera showing up in these distinct zones and areas with various fossil taxa then present in between. It's also showing a lot of these fossil taxa, again, this being, whoop, lost my light, so I'm gonna do stuff.
I think your computer adjusted, so it's all right.
Yeah, well, we'll just continue. It'll just be a little bit darker then.
Yeah.
That'll work. So, again, we're getting these fossil taxa coming out as not within a monotypic effect of any of the genera, which seems to be completely fine. It just suggests that either we need more information for a lot of fossil taxa within these groups, or it means that we're getting a lot of lineages that went extinct that didn't make it to basically the modern day, which is not overly surprising. You'd expect extinctions of some of these lineages and not all of them to make it through. So if you look at that and you compare the morphologic information again to the genetic information and the genetics that we get currently as far as the relationships are concerned, you'll see some similarities and a lot of the same relationships. But again, one of the big differences is that you get the painted turtle coming out as a very different spot here, being basal within this group and genetic speaking higher up, and not much higher up, because basically the only more basal group is the chicken turtles in yellow here versus the painted turtles down in orange, but you still see a distinct difference as far as where their grouping is concerned. And then as a note, you'll see up here a little bit higher, you'll still see the effect of the modern genera for the amidines coming out. And again, part of the difference is that oftentimes you get clemmies coming out as sister to terrapene, and morphologically speaking, it seems to shift to a more basal spot within that group. And so as we look at the gray fossil site, you get a wide turtle fauna, you get a large range occurring. And so you get things like Trachomys, the slider turtle, but you also have a lot of other turtles living within that, and you get things like Chrysomys, the painted turtle, Terrapene, the box turtle, and something either very similar to or almost the same as Emydoidea, the Blanding's turtle, within the amidids at the site alone. In addition, of course, we're getting other turtles within the fauna itself, including chelydrids, penosternids, and multiple types of tortoises or tessudinids. And this is showing us that we have a wide variety of turtles. It's not surprising given the environment, but also the degree of variability of the environment. So the idea that not only is this just a pond-like environment, but that you also get a wide variety of more open habitat, potentially similar or close to this, gives the ability to have a much wider range of these turtles present. And so even though most of these are going to be very aquatic and staying in the water, you're going to have other varieties that are either semi-aquatic and only spending a little bit of time in the water, or basically fully terrestrial. And maybe that's why, of course, some of these more terrestrial forms, like the tortoises, are more rare within the turtle fauna compared to a lot more of the aquatic animals that we're seeing. One thing,
not only rare, but also very different in their preservation.
Yes, absolutely. And so part of one of the things that might be also abnormal, I suppose, is the fact that when we think about turtles today, especially these turtles that I'm showing, at least these groupings of turtles, because these, of course, are all modern pictures, you're looking at things like, hey, painted turtles are very, very good at living in smaller ponded environments and basically water that doesn't move. And so are snapping turtles, so chelydrids in general. But if you look at things like sliders, for the most part, when you think about where they actually live, they either live in much larger areas or think of like slow moving rivers. So the water is at least somewhat moving, not extreme, not fast moving rivers, but it means that basically the environment that you would, in this environment, you would expect things like chelydrids and chrysomes to be the most common turtles. They should enjoy the environment more than any of the other turtle species shown here. And even oftentimes kind of sternids are oftentimes a little bit more murky, mucky water and things like that. But it seems that the Trachemes seems to be the most common turtle present within the actual turtle fauna. And you seem to have the largest sample size of that, which is either suggestive of a difference in habitat preference for at least this species, or it might mean something else is going on that we're not really having a really good handle on. So there might have indications of what's happening with that, either for the species in general, or maybe what the species is actually preferring within that.
All right, thank you. If you could go ahead and start finishing up here, we'll move on to questions in about 2 minutes. Just a reminder for people to send in their questions.
Certainly. Actually, I think that actually works out really well as far as timing is concerned. So currently, I'm also working on the other amidids, and it's interesting in that with the Trachemes, we determined it was a new species within a modern genus, and the same thing was done with the Kinosternid. So it's interesting in that in doing further work on these fossil amidids, it seems like there's a good chance that we're also looking at new species within these fossil amidids. So all of these turtles are different from everything else we're seeing. And it's interesting that most of these seem to be representatives of modern groups, essentially. At least we're looking at crown groups within all of these turtle taxa other than testudinids. Now, testudinids are very fragmentary, so we only have a little bit of information for the most part about them. However, they with the modern genus mainly being Gopherus, or at least Gopherus-like tortoises in North America today, these are not fossil Gopherus, at least from what we can tell. So they're different, they're members of fossil groups and fossil genera within the tortoises themselves. And so it's interesting that the more aquatic versions seem to be basically more crown groups, whereas these tortoises seem to be made up of distinctly more fossiliferous, or at least fossil genera and groups that are now extinct. So it'll be interesting to see what comes about with more material and more study from a lot of these other turtles, what we end up determining as far as where they end up lying. So with that, tons of people to thank, and this is, very, very small, but,
All right, perfect. Thank you, Stephen. We have lots of questions coming in, and David will jump in with some Facebook questions.
Sure thing. So as a reminder for everybody watching, now's the time to send in questions. You can go ahead and leave your questions in the comments in the Facebook, on the Facebook video. And again, if you can't leave a comment on Facebook for whatever reason, head over to the Gray Fossil site on Twitter or Instagram and leave a question there. Steve, I'm going to start with a very straightforward question from Greg McDonald. Has Trachemes haugrudi been identified at any other sites besides Gray?
It has not. It's very similar to a fossil taxon from Florida named Trachemes inflata, which happens to be one of the sister taxa to it. And so it's very, very similar to that. And there are potential similarities with things like the fossil site in Indiana, although there's a better chance of actually potentially finding other turtles from Gray there than the Trachemes species. So currently it's only known from the Gray fossil site with a very large sample size, albeit from one locality.
Gotcha. Our friend and former co-student Aaron in the comments has pointed out that there are also tons of Trachemes down at Montbrook and welcomes you to come look at the Trachemes down there.
I am all for it. It's certainly possible that we could find it further down there. And Inflata is actually a bit older. It's late Hempylian. So with this being a little bit younger and more similar in age to things like Trachemes platymarginata, which is definitely different, but of a bit similar age, we could certainly find this species working its way further south. I'd love to be able to do that.
Yeah, Inflata is ridiculous in terms of its thickness of the shell. Just silly in the way that it looks almost.
Yeah.
Have you seen anything like that or trend toward that in ours?
I mean, really, for ours, the main portion that gets inflated is the very front of the shell. So you're looking at the peripherals and the nuchal, the very front edge of the shell gets really inflated, but a large portion of the rest of the shell does not get nearly as inflated versus Inflata, where a large portion of the shell all over is inflated. And even the parts that are inflated, like the nuchal or Haugrud, is not to the same degree in any of the specimens as it is in Inflata.
All right, moving on to another question. Earlier in the talk, I believe you had mentioned similarities between European and Asian and North American taxa. Mason asks, how did the European genus get there? Were they at one time more diverse in Eurasia?
A very good question, and we actually don't have a really good handle on that. With the fact that the turtles would have been first evolving in the late Eocene, then the fact is you have to assume that they are not making it over to Europe until much later, and probably when it's very, very warm before it starts cooling down. Because once it cools down, you get rid of a lot of those northern pipelines that a small turtle would need to get over. I mean, small turtles are not going to move very much. We know that modern aminids oftentimes, especially depending on the environment they live in, move very, very little. If you are in a waterway, you can follow the waterway a lot easier. But again, these are not, some of these turtles are saltwater tolerant slightly, so you can kind of get into saltwater a little bit, but nothing to go the whole way over there. So we don't have a good handle on what's happening, and we don't have a good fossil record of these turtles in Europe at this point in time. So it's not as easy to understand how they're changing and then therefore what they're fully coming from. They seem to be most closely related to the Pacific pond turtle, so the one in the western US. So that suggests that, of course, they're making their way over from, they're probably making their way over, of course, from North America across the bridge and over into then Russia and continuing on into Europe. But we also have no fossil record of them then in Russia or Asia. And so there's a big jump as to how they're getting over there.
Looks like Chris has a question.
Oh yeah. Hey Steve, fun talk, and it's always fun to think about turtles. And as a person who kind of holds a lot of turtle taxonomy and turtle paleontology at arm's length, I notice every couple of years there's this paper that comes out that says, oh, we have a completely different understanding of how the turtle got its shell, how they're related to other reptiles. And we have a question from Charlie Bruce. Can you tell us more about the most up-to-date thoughts on turtle evolution and how they relate to other reptiles? Can you do that in 30 seconds?
That's what I was going to say. That's a whole show.
We can give it a shot real quick. So the basic idea for turtles is that we still, a lot of it depends on who you think they're more closely related to as to probably where they're really coming from. And so you have the idea of whether they are kind of more similar to lizards and squamates in that sense, or whether they come from something much further back. And then therefore, are they losing elements of their skull as far as anapsids are concerned, or are they losing multiple, or are they not? So all of those kind of things. We still don't have a really good handle on that. We think that right now the genetics seems to tell us one thing, and morphology seems to tell us something else. And so I think it's more kind of coming to grips, or at least some kind of agreement between those two things as to where the real relationship ends up being, and therefore where they actually end up evolving from. The basics are that it's still, there's two major camps, I would say. and both camps have legitimate points as far as what they kind of consider. So we still are waiting for more information to really fully decide which one is correct or not. But both sides can be very forceful as far as what their points of view are. And so that's probably why you get multiple things coming out from one group and then the other group every couple years, determining that this is more correct than A is more correct than B and B is more correct than A.
All right, we've got a question here from Ethan, our friend Ethan, who asks, does North American turtle diversification respond appreciably to environmental events in the Cenozoic?
So based off of newer research that was occurring after this, it seems like there's a very good chance that turtle diversification is dealing at least partially with some major events that are happening. And part of that you can see in the fact that when you get large diversification in some of these turtle groups, it's happening after some major events. So when you look at the Cenozoic, oftentimes you get things such as grassland evolution, and you get a lot of open environments and cooling during the Oligocene. But you also look at things such as the mid-Miocene climatic optimum. And so you're getting a lot of warmer events. What you end up seeing is shrinking and to a point, extinction of some groups and then diversification of other groups. So the short answer is yes, we do seem to be seeing that. And current research, including research on some of the turtles from Gray, is helping kind of convey some of that information to us a lot more clearly.
Stephen, I have a quick one for you. If someone was going to ask you, how do you distinguish Huggard's slider turtle from other slider turtles? Is there a simple and short answer of a character of the skull or something like that, or a couple of characters that would lead you to an easy way to identify that?
So the easiest ways to look at that are going to be, there's a couple features of the shell and a couple features of the skull that really help convey that. And narrowing it down, if you really look at the proportions of the nuchal, the front bone within the shell, you get some clear differences, especially with the degree of serration, so the degree of the pointing and indentations within it. You also get clear differences within those same indentations, so the serrations or the notches within the front and back of the shell. So the rims of the shell itself are both very important for that. And then for the skull itself, you're going to get features within the lower jaws, especially features of the triturating surface. That's the surface that they actually chew on, that kind of goes flat within each other. And then the elongation and lack thereof and the morphology of the squamosal, so that front piece on the back. So if you're looking at the shell, the skull right here, the morphology of this squamosal also is very key to determining what you're looking at.
Now, are there parts that you're missing that you really wish you had that would be quite telling?
So parts that would really, really help out would be parts of, with the skull itself, because it's in two pieces, we get a view of the interior of the skull, but we also lose portions of the back of the skull that would be really useful and helpful. There's also features of basically what you consider the nose of these turtles, the nares, so the opening up here, that we're missing, that would be very telling to easily convey both the similarities and differences between these turtles and very similar turtles. So parts of the front of the skull and parts of the very back of the skull would be very useful if we had them. But premium material is rare enough that even that would help tell you the type of turtle, and it would separate it from a couple different turtles. But most turtles, again, including Amidids, are known from the shell itself. And so the shell is going to be the most important thing. And because we have the entire shell, we have the ability to thoroughly explore how the shell compares with other ones.
All right, keeping it on the topic of the gray fossil site turtles, on Facebook, Leah asks, given the depth of the deposit at Gray, do you expect over time to find ancestral taxa to Haugrud's turtle, or do turtles evolve slower so you would predict to see the same taxon throughout the deposit?
So my initial thought would be that it's possible to find turtles simply for the fact that they can evolve quickly, but a lot of this depends on who you're asking. And so especially with Trachemes, when we look at this genus, I heard kitty, when we look at trachemes, oftentimes the thought currently is that all Pleistocene species of trachemes are all a single species, and they all represent the modern main species within North America, Trachemes scripta. And so the thought is that because you see so much variation in the modern, and because they're not evolving quickly, they're all the modern species throughout the Pleistocene. And everything we found seems to say that. But if you take it that variation is not as extreme, then you may take it as, well, if you take it that the variation is not as extreme, then you're going to take it as these turtles are evolving more quickly, and so you're seeing minor differences are showing you differences in these turtles. For trachemes, an ancestral version of Haugrud may be very, very similar to what we're seeing, and if we go off of similarities with things like Inflata being an older sister species to it, or an older sister taxon within the same group as far as this polymorphic group or polytypic group, then what you're going to see is simply an inflation of some elements for Haugrud, but that's going to be the main and only difference you're going to see in potentially older versions lower down. Everything else would be pretty similar, at least from what we can tell. Part of the problem may be that we don't have a complete shell of Inflata, so it's going to be really hard to tell what the entire shell would have been like. But going off of that, you would assume that there would be potentially minor differences in some areas, including things like inflation of the shell, but that might be all you're going to say.
I'll just jump in and comment real quickly, and maybe Chris is also, but right now it seems that the deposition that we're getting at Gray is relatively short-lived, geologically speaking. And since we're not seeing yet, at least, a lot of changes in the mammals, which can, especially the small ones, can have a much faster turnover evolutionarily, we might not expect it with the turtles.
Yeah, kind of sticking on the same, topic of gray turtles, Steve. We have like this great, one of my favorite specimens in the Great Fossil Site is this tiny little mud turtle that has been crunched on. We know something is eating it. And I'm just wondering, with the trachemes, do we see any, what is eating the trachemes? Do we see bite marks? Do we see crunching on them? That sort of thing. Have you seen anything like that in your work?
So, I mean, while oftentimes you can see turtles today, oftentimes have a lot of bite marks. Oftentimes the bite marks you're seeing or going to be seeing are predation events that mostly fail. And that's because the main thing that you're going to find eating these turtles probably are the alligatorids, right? So the alligator at Gray would be the main culprit you would think of. You're not going to see turtles being eaten by a lot of much larger animals. They're going to cause a lot of damage. Sometimes you'll see predation on turtles from smaller carnivores. That could be things like raccoons today, but it could have been things like the red panda at Gray. But then you would not see that on the shell at all. You'd only see that on postcrania. And while we do have postcrania, we still don't have a ton of postcrania to hopefully get at something like that. For other turtles, when you're seeing broken shells, oftentimes you're seeing shells that are kind of bitten by an alligator or alligator-like animal. And so you're getting tooth punctures and things like that. But again, those are failed attempts. Oftentimes, if the alligator gets the entire thing, they don't suck the turtle out. They eat the entire thing, shell and all. And so they crush it up, and with their digestive systems, most of that gets broken down. You don't really get almost any of the remains from that. So while predation on turtles is common in the fossil record, and we would expect to see that with the kind of the fauna that we have at Gray, at least from what I'm aware, we don't have any definite signs of that yet. Oftentimes, a lot of the holes and punctures we're seeing on the shells at Gray are actually due to infections and bacterial growths underneath the scutes that cause holes and openings within the shell itself. And this is common within a lot of aquatic turtles, and we're seeing that on a lot of specimens there.
All right, I've got a question here from Facebook about modern turtles that I think you'll appreciate. This is from Janice. Because I know, Steve, that you also enjoy going out looking for modern day living turtles. Mm-hmm. Janice asks, I've read that a box turtle should not be removed from its home territory, otherwise it will wander aimlessly the rest of its life. Is this true, or is this an urban myth to stop people from taking them home?
So box turtles, as most turtles in general, will have actual home territories that they focus on, and they rarely leave those territories. So in removing a box turtle or any turtle from its normal home habitat, it will potentially lead to difficulties and problems. However, some turtles will wander a long way for a lot of other reasons. Oftentimes, male turtles will wander a very long distance simply for the fact of looking for mates. And it happens a lot. It's one of the main reasons that you'll see turtles wandering across roads and things like that. And females will oftentimes do this in looking for nesting sites. So good areas to actually have nests that might not be in their home territory. Now, if you remove a turtle, it will lead to difficulty. It won't wander aimlessly. It will wander until it finds another suitable habitat that works for it. But that might take a while, and you might release it into an area that doesn't have any suitable habitats for a very long distance, and so they might wander a very long way. So for the turtle's sake, it's best to either not remove it or put it back to where it originally was, because you never know what the result will end up being for that turtle if you randomly release it somewhere else.
And you also don't want to take a turtle and have that turtle in your home for a while and then put it back into the wild. That's another big no-no.
Yes. There's lots of difficulties and problems that can come for that turtle in those situations. So really it's best to enjoy the turtle where it is without picking it up or taking it or removing it.
Solid advice. We are getting near the end of our time here, but I've got another question from Charlie, which is about a totally different group of turtles we haven't talked about yet in this talk. Charlie asks, what is the fossil record of side-necked turtles like? Were they more diverse at one time?
So side-necked turtles actually have a really good fossil record, just mainly not here. If you look at places like South America and over in the Old World, they actually have a very good fossil record. Not as extreme or a complete, I would say, as some of the other groups, even including things like Amidids, even though the Amidids are very fragmentary as far as their normal fossil record is concerned. But they were certainly more diverse in the past, and we have a much better fossil record of them in other areas. But we still don't know nearly as much about their evolution as we could, because oftentimes the fossil record is still so incomplete and inconsistent. So we're still looking for a lot more, but certainly more diverse in the past.
All right. Blaine, I think we're right about at the end.
I think we are. Well, thank you again, Stephen. It was a pleasure. We all learned a lot about turtles today.
No problem.
All right. We'll see you all next week.